Molecular engineering. So today's topic is molecular engineering. I'll talk about it in two sections. One is introduction and the other part is application. So I'll start with introduction. What is molecular engineering? It is the branch of science that deals with engineering of the molecules to do something useful. So it's an interdisciplinary science that brings together different kind of sciences like chemical engineering, mechanical engineering, material sciences, bioengineering, electrical engineering, physics, chemistry, all together to make molecular engineering successful. So it's an emerging field that deals with designing and testing of molecular properties at microscopic level and assemble better material systems and processes that suits our desired functions. So in this approach we move from microscopic level to macroscopic level. This kind of approach is called as bottom up design or bottom up approach. So molecular engineering, it also overlaps considerably with nanotechnology because both are concerned with the behavior of material on the scale of nanometers or even smaller. So molecular engineering, it's a dynamic and evolving field that is based on molecular principles, and there are n number of applications of molecular engineering like immunotherapy, synthetic biology, quantum engineering, energy storage, etc. So it ranges widely. Its potential application ranges vastly from energy to healthcare to electronics. So molecular engineering, it involves both computational tools, experimental method, or even both. So basically, what is molecular engineering? It is dealing with the creation of molecules, with the creation of molecular and development of new products from them, and these molecules may be existing in nature or these may be completely synthetic, which we are altering at the microscopic level using molecular engineering approach. So it requires a lot of sophisticated technologies and tools and equipments to do this molecular engineering. For example, we use electron microscopy, particularly scanning tunneling electron microscopy, atomic force microscopy. So all these techniques, they are used to do molecular engineering of biomolecules or even the organic molecules. So this is a graph that is showing a correlation between the size of the molecule and the complexity of the system. So as we move, this level is representing the molecules are designed using this molecular engineering approach, then they are self-assembled, and that leads to a particular system or a novel system or a device system functionality. So there are three levels during molecular engineering. One is when at the microscopic level a molecule is designed or its surface properties are altered using the principles of molecular engineering. Then these are assembled, and then that leads to the development of particular system. So we in molecular engineering we move from macroscopic level from here to microscopic level to the molecular level, and then we alter the properties at macroscopic level. There is a correlated change in properties, change in bulk properties at macroscopic level as well. So what we are doing, we are moving down in size. We are moving from microns to nanometers to even angstroms. So this is the diagram that is explaining the molecular engineering approach. Okay, this picture is depicting here these are the molecules, these are the molecules which have been designed using molecular engineering. So this is the first step, molecular designing at the microscopic level. Then these molecules tend to self-assemble with unique properties, and they get assembled. First step, second step, and after assembly we leads to the development of a novel of a particular function or a device or a process. So there are three main steps in bioengineering or in molecular engineering. First, we design the molecule. We alter the properties of the molecule. It could be physiochemical properties, it could be the surface properties, any kind of optical properties. So these properties are altered. Then they are made to self-assemble. They are assembled, and after assembly, a novel process or a system is developed from these macroscopic particles. So this is the rationale, this is the idea behind molecular designing or the molecular engineering. So it is molecular engineering there is a direct correlation between the development of molecular design approaches towards the system of higher complexity. Molecular engineering, it deals with design, testing of molecular properties, behavior and interactions in order to assemble better materials, systems and processes suiting specific functions. So this type of approach, in which observable properties of a macroscopic system are influenced by direct alteration of a molecular structure, is called bottom-up approach. So what is bottom-up approach? In which the properties of a macroscopic system is being altered. There is a direct correlation between the macroscopic properties that can be altered by altering the properties of the microscopic system at the molecular level using this approach, molecular engineering approach. So again, this is a diagram that is showing the interrelation or the sciences that are all contributing towards the development of molecular engineering. So it's a highly interdisciplinary branch of science. Okay, a little about the history of molecular engineering. So this concept is not very old. It was first mentioned in 1956 by Arthur von Hippel. He was the first person who gave the concept of molecular engineering, and this concept was reinforced by Richard Feynman in 1959, who quoted that there is always a plenty of room at the bottom of, there is a plenty of space at the bottom of the which means that there is always a possibility that we can design something new, we can create something new if we go from macroscopic level to microscopic level. If we go to the molecular level of a system, we can always there is always a plenty of scope to bring any kind of alteration in the macroscopic system in the bulk properties of the macroscopic systems. Then after a while in 1980s, this Drexler was there, who showed the correlation of this molecular engineering with nanotechnology. And in 1977, Alan J. Heger, he discovered polyacetylene that led to the development of organic electronics. He gave organic LEDs, light emitting diodes, and flexible solar cells. So also with the advent of colloid science and surface science, there is a mention of Erwin Langmuir and Catherine Blodgett, who also contributed for the development of molecular engineering. So after a brief history of molecular engineering, next is the techniques and the kind of instruments that are used in molecular engineering. So with the advent of electron microscopy, particularly the Knoll and Ruska, they are credited. Knoll and Ruska, they are credited with the invention of electron microscopy, and this Gerd Binning and Henrik Rohrer, they are credited for the design of scanning tunneling microscope. So with the development of this scanning tunneling microscopy, that gave actually the birth to nanoscience, nanotechnology and biomolecular engineering. Okay, these are the various molecular characterization techniques. After microscopy, these are the techniques by which a molecule is characterized. It includes this MALDI, matrix assisted laser desorption ionization spectroscopy, NMR spectroscopy, size exclusion chromatography, Raman spectroscopy, and TOF, time of flight, secondary and mass spectrometry. So these are the all spectroscopic and techniques that leads to the characterization of molecule. So all these together with microscopy, they lead to the birth of molecular engineering. So apart from these synthetic molecular engineering technique that includes DNA synthesis, nanoparticle synthesis, organic particle synthesis, peptide synthesis, polymer synthesis, they all together contributed to the development of molecular engineering. So I'll take application part later. This is all that has been engineered, like it's molecular engineering. So what kind of molecule exactly we are dealing with? It could be a protein, it could be an enzyme, it may be enzyme engineering, that is again a protein. It could be drug designing, or it could be metabolic engineering, tissue engineering. So all these come under molecular engineering. So I will take only protein engineering in detail. So with the advent of this site-directed mutagenesis in the gene and availability of computer-aided molecular modeling tools, it is possible to engineer the protein. These are the basic steps of protein engineering. First, the protein sample is extracted or prepared from the concerned tissue. Then its 3D structure is determined using X-ray crystallography or NMR, nuclear magnetic resonance. Then that 3D structure is displayed with the help of computer graphics and designing tools. These 3D structure, it is displayed with computer graphics, and there using the same tools we design the structure, we alter the structure, 3D structure that suits our needs. For example, we are designing a drug, so we will design that kind of drug that fits better to the ligand or the target molecule. So there is a change in design. So now that the change in design is made, then this design, it is like it's the desired DNA sequence is obtained. There are two ways to get this designed DNA sequence. Either it is synthesized chemically, or there we can go for mutation in the gene. So then novel means new gene is introduced, that we obtain either by either of these two techniques, that is introduced into the expression vector and then into the host. This desired protein, it is extracted, purified and characterized biochemically. Now after biochemical characterization, if this engineered protein, if it is matching our requirement, then it is okay, we will proceed. If it is not, then we will again repeat the cycle. So this is protein engineering. It is based on knowledge based modeling. This kind of approach is called as knowledge based modeling. So in this enzyme engineering, drug designing, and this peptide vaccine development, all these share the same steps as we have I've discussed over here. So this is all about the molecular engineering. What is molecular engineering? Its introduction, its history, how it really came into light, then its various kind of techniques and instruments that we use for the molecular engineering, and finally the application part. What are the main applications of molecular engineering, why we do this? So application part I will take later. This is all about the introduction.